Okay guys, welcome back to Biology AP. Today we are going to be talking about those macromolecules that we hinted at in the last lesson. So if you'll recall, we learned a little bit about what organic chemistry is and how a lot of these compounds that make up living things actually contain carbon, which that's where the organic chemistry bit comes from. So today we're going to cover those big overarching groups of macromolecules, and we're going to focus today just on carbohydrates and proteins. We'll be covering the other two in the next video. So let's get started. Carbohydrates. So carbohydrates include sugars and polymers of sugars. So sometimes these can be quite long. Starches are just long chain polymers made out of sugar. Oh, so fun fact, if you want a science thing you can try at home, if you have saltine crackers, scratch a little bit of the salt off the top and sit it on your tongue, make sure there's lots of spit on your tongue, and wait. And eventually you're gonna break down that starch into sugar because there's enzymes in your spit, and it will go from being, you know, a little bit salty to actually being slightly sweet. So that's a thing for you to try. So carbohydrates generally contain a carbonyl group and many hydroxyl groups. So again, I'm filming at the school, so I don't have access to my iPad. But if you will look over here to the side, you will see that we have that covalently bonded carbon and oxygen. So that's a carbonyl group. Remember, it has to have a COOH to be carboxyl. So this is definitely carbonyl. And there's lots of hydroxyl groups. So all of the O's and H's that are together, that's a hydroxyl group. So carbohydrates are comprised entirely of carbon, hydrogen, and oxygen. Monosaccharides, mono means one, so those are simple sugars. So they come in a formula with multiples of the unit CH2O. So it's always a 1 to 2 to 1 ratio. So 1 part carbon to 2 parts hydrogen to 1 part oxygen always. So you may notice there's a carbon, but then there's H2O, and that looks suspiciously like water. So this is why it's a carbohydrate. Remember, hydro means water. So it's carbon with water thrown in, and somehow it tastes sweet, because science is weird, y'all, okay? It's weird. So the most common is glucose. If you know anyone with diabetes, you've heard all about your blood glucose and your sugars. That's because your body has a tricky way of turning any carbohydrate you eat just about into glucose. So that's why diabetics can't have a lot of starch, because like we said a minute ago, a starch is just a whole bunch of sugars crammed together. So it's super easy, if your body gets some starch in it, it can just chop up that starch into a whole bunch of glucoses. So you actually flood your bloodstream with sugar, and you can go into, you know, some negative diabetic effects. So carbohydrates are most oftenly used as nutrients and fuel for cells. Sorry guys, my hair is in my face, the air conditioner came on. So nutrients and fuel for cells. So it's used in cellular respiration. Guys, this is, don't worry too much about this right now. We're gonna get to cellular energetics. That is a problem for another day. Yeah, but it's coming. So cellular respiration, and it can serve as building blocks for amino acids or as monomers for di and polysaccharides. Di, how many is that? Dice are how many? There's 2, right? So disaccharide it's not quite a polymer because there's only 2 of them. So anyway, let's keep going. So like we said, a disaccharide is 2 monosaccharides joined together by a covalent bond. Sucrose is probably y'all's favorite because that's table sugar. Yeah, straight up table sugar is sucrose. So the monomers that go into that are a glucose and a fructose. So glucose, you know, that's your basic like blood sugar. Fructose, that looks a little bit like fruit, right? So that's generally considered to be a fruit sugar. So we actually commonly get sucrose from sugarcane. So we distill it or whatever from a plant. So that's where the fructose comes from. So the plants transfer carbohydrate from the roots to the leaves in the form of sucrose. So, okay, if you know anything about photosynthesis, you're probably confused right now. So remember that photosynthesis makes glucose, okay? So the glucose gets sent down to the roots. They put it together into sucrose and store it, and they can release it as the plant needs it. We'll talk about it later, but plants have to use that sugar sometimes. Like they need food too. Plants got to eat. All right, polysaccharides. So poly means many, okay? There's a whole lot of other words with poly in it. It's late in the afternoon, guys. I really can't think of a whole lot of them that are necessarily school safe that I want to put on YouTube, but y'all are smart, okay? Polysaccharides, so it's a polymer with many sugars joined together with those dehydration reactions that we talked about. Oh, good, I just thought of one. A polyglot is someone that speaks many languages. So a polysaccharide, many sugars. Okay, so polysaccharides are quite often storage, but sometimes they're structure. So the storage ones, like starch, think like a potato. I don't know if you've seen a potato plant before, but it looks just like a plant on the top, and you pull it up and it's got these big, the potatoes are on the roots. So that's where it's been storing all that starch for the plant to use later. It's just, we're really rude and we yank up the plant and cut off its roots so we can eat them because French fries are delicious. Sorry. So animals also can store carbohydrates as glycogen. Well, I had a student that absolutely got mad at me when I tried to explain to her that when we refer to animal cells, that's people. People count in that category. She's like, no. What kind of cells are people made of? Like, sorry. So this goes for us. So what happens, for example, let's say that you really hated the sack lunch in your static group and you decided to chuck it in the trash can. Well, you still have to breathe and blink and sit upright and all that. That takes energy. So you don't have a whole lot of blood sugar because you threw your breakfast in the trash can. So then your body is able to send a message to your liver, and it starts breaking apart that glycogen to put some sugar back into your blood. We're going to talk about cell signaling later in the year, and we'll talk about how they do that. Okay, so glycogen is stored in liver and muscle cells. For my athletes out there, that's actually why your muscles burn when you exercise, is you're not getting enough oxygen to burn that sugar cleanly into just whatever. So you ferment it instead and make acid, and that lactic acid is what makes your muscles burn. I'm getting sidetracked. I apologize. Okay, structural polysaccharides. So structural is like the support beams, like, you know what a structure like a house is, it's like the supports and all that. So cellulose, that forms cell walls, the hard bit of a cell wall. And Google Slides, okay. So chitin does the same thing, but in arthropods. So that means like crawfish, crustaceans, sorry guys, insects, they're built the same way. It doesn't list it on here, but actually the cell walls of mushrooms also have chitin in them, which weirded me out when I first learned that. Okay, we're gonna skip. Well, I guess we could talk about it. So you are given an unknown monosaccharide to identify in the lab. Remember, mono means one. The only clue you're given is that it has 4 carbons. You, being an excellent AP biology student, figure out the formula and name immediately. So what is the unknown monosaccharide? So remember that it has to be a 1:2:1 ratio, right? So if you want to take a minute and figure that out, if we have 4 carbons, what are the rest of them going to be? Okay, so since there's 4 carbon, that means I have to have twice as much hydrogen, or 8, and then 4 oxygen. To be perfectly honest, we never learned about naming these when I took biochem, so I can't explain to you how they got this name for it. but there's no excuse not to know how to get the chemical formula, okay? Like, I'm not expecting you to know that it was 3-O's or whatever shenanigans, but you should be able to figure out the C4H8O4 bit, okay? Okay. Proteins. Okay, so proteins, we start with one amino acid, okay? We can join 2 of them and make a peptide. We can have a chain of them and make a polypeptide, and then we can make a large protein. So molecules consisting of polypeptides, polymers of amino acids folded into a 3-dimensional shape. Okay, let's let that sink in for just a minute. Okay, so proteins are commonly CHON and sometimes S. So mostly for proteins, I'm really looking for a carboxyl group quite often because there's a reason they're called amino acids. And I'm looking for nitrogen. If you see phosphate, you're way off. You're not in proteins, you're in something else. Okay, so shape determines function. I wish I had my iPad on because this is so important. This is a main idea of this lesson. Y'all write it down, underline it, type it big, whatever you're doing. Shape determines function. If I change the shape, I change the function. Anything that changes that shape is going to affect what that protein does. I know I'm really intense right now, but I need to get it in your brain, okay? Get it in there. Don't put it in long-term memory. You're gonna need it. It's gonna be super important and a recurring theme in biology AP. Okay, so here's what I was telling you about amino acids. So they've got an amino group, like I told you. They've got a carboxyl group, like I told you, and then a variable side chain that could be different kind of foolishness, okay? So let's look at some examples. Alanine is one of our 20 amino acids. You are not going to be required to memorize the amino acids. Oh Lord, that would be way too much, y'all. I am not that evil, okay? You'll have a chart. So this one's alanine. So we've got our H2N, so our amino group. Go away, okay. You've got your carboxyl group to the right, that COOH, the COO group. Oh gosh, please don't mock me. And then the rest of it is considered a side chain. Then we can look at glycine, right? So they tried to color code it, the little tan box, that's your side chain. So it's a CH3 right there, right? But in glycine, it's just an H, that's the difference. Okay, it's going to affect some stuff. So let's keep going. Oh, go away. So they're just trying to tell you it's the unique side group. Okay, so each amino acid has a unique side chain. Okay, so unique aspects of the amino acid are based on the side chain's physical and chemical properties. Side chains can be grouped as nonpolar or hydrophobic, polar, which are hydrophilic, and charged or ionic, which are also hydrophilic. Okay, so there's some of you probably like, Miss Vance, you're speaking Urdu again. Okay, so nonpolar, okay, hydrophobic. So phobic, like if I'm phobic of spiders, I'm afraid of it, right? So it's nonpolar, it's afraid of water. It won't dissolve in water, okay? Polar is hydrophilic, meaning that it loves water. So I'm trying to think of school appropriate words. Again, guys, I don't know why they let me make videos this late in the afternoon, okay? I'll be real. So like a technophile, they love technology, okay? Hydrophilic, that loves water. It dissolves in water. Ionic or charged compounds, they will also dissolve in water. I think we talked about dissociation when we talked about the solubility of water. So those things will definitely be hydrophilic. So depending on how that side chain interacts with water depends on what it's gonna do, okay? Okay, so peptide bonds. I can promise you, you're gonna see this on a test. So to form a peptide bond, the carboxyl group of one amino acid must be positioned next to the amino group of another amino acid. This is a dehydration reaction. We're going to use those for a lot of stuff, okay? But this is specifically a peptide bond because I'm joining amino acids, and instead of doing some cute stuff with carbon and oxygen right here, carbon and nitrogen are going to form a covalent bond and join them together. So it is a peptide bond, okay? Polypeptides. So we can have lots of amino acids linked by peptide bonds. Each polypeptide has a unique sequence of amino acids, okay? So we'll focus on that for a second. When we make a protein, it's like beads on a string. We have the initial sequence, right? 1, 2, 3, 4, 5. And then directionality. How are they put on there? Okay, so each end is chemically unique. One end is a free amino group. One end is a free carboxyl group. Because you think it's repeating, so it's got to have an end somewhere, right? Okay, so the sequence of these amino acids determines the three-dimensional shape. The way I think about it, I don't know if you can see this on the camera, so let me unshare for a minute. Stop share. Okay, so you see like my lanyard, right? It's perfectly straight. So this is like my primary structure, right? But what we're saying is you can twist it and keep twisting, and eventually, see, it knots in on itself. Proteins do the exact same thing, okay? So let's reshare so that we can keep going. Okay, so the sequence of the amino acids is going to determine that. All right, remember, shape equals function. Super, super important. When a polypeptide twists and folds because of the R groups interacting, it forms a protein. Remember, some of them are going to be hydrophobic and they're going to want to hide like inside the molecule. So they're not going to want to face out, and that's what makes it fold and twist like that. Okay. Function of proteins includes antibodies. We've been talking a lot about that in the news because of the corona. They're looking at antibodies because they help people recover from diseases, but they're made of protein. Enzymes, they carry out chemical reactions or assist in creating new molecules. We're going to talk more about those later, but those are also proteins. So they can be messengers. They can be structural, like your muscles. They can transport things. We're gonna talk about transport proteins till y'all are sick later, so just be prepared for that. Oh, sorry. Okay, levels of protein structure. So primary, the linear chain of amino acids determined via genes. So like your DNA, it dictates the secondary and tertiary form. So secondary coils and folds due to hydrogen bonds within the polypeptide backbone. So we can have a pleated sheet. So the hydrogen bonds between polypeptide chains lying side by side, or an alpha helix where they twist. So when I was showing you my lanyard, would that have been a pleated sheet or an alpha helix? Be an alpha helix, right? Because it coiled around. So tertiary is 3D folding due to interactions between the side chains of the amino acids. Okay, so the covalent bonds form between the, all right. So this is when my lanyard knotted up. And then quaternary is when there's more than one polypeptide, how they nest together. And only some proteins do this. Ribosomes definitely do this. Hemoglobin does this. So I'm going to show you again, right? We're going to stop sharing. Okay. So trying again, primary structure, right? So the straight line, primary structure is like one-dimensional, right? And then I coil it. Remember, this was an alpha helix, right? So this is my secondary structure. And then how it coils in on itself, right? This is my tertiary or three-dimensional structure. And I don't have another lanyard lying around, so like how it nestles together with something else, that's quaternary. Okay, I know that's a lot. Share screen. Okay. So all 4 levels of this is going to be important, and it's also going to be on your test. So gird your loins. Okay, so guys, we made it. Okay, so real quick, I'm going to show you what I want you to do today. I know we only covered the first half of macromolecules, and y'all are like, Miss Vance, stop going back and forth. I'm so sorry. Okay, so hold up. Okay, so you're going to be working in your OpenStax textbook today. Okay, so I'm at OpenStax. Oh, y'all, hold on. Okay, y'all got to forgive me. I'm figuring it out. Okay, so OpenStax. Now you can see what I'm seeing, and I'm not talking to myself. Okay, so you'll have this link to Biology 2E. I think the way I've got it set up, I can give you a direct link. So you're going to have a reading guide for chapter 3. It's going to cover all of this, right? 3.1, 3.2 through 3.5. It's not super long, but I do expect you guys to do it. It'll help get you ready for the second half of this. Yeah, so make sure you're getting that done and submitted. We're just using this online textbook. I think at this point we've used it once before. You have to forgive me. I have to plan these lessons 2 weeks in advance, and sometimes I get lost. I think we've already done this by this point though. So yeah, this is our textbook. It's free to view online. You can view it from anywhere. It's pretty cool. So guys, good luck. Let me know if you have any questions, and I'll see you in the next video. Take care, guys.